Okay, good morning, everyone. I guess we're live, but let me quickly check everything. Everything is working, and then we're gonna start. Okay, I can hear myself on YouTube as well, so I guess we can get started. All right. Turn on my camera also. All right, I guess you can see me as well. So you can ask your questions, by the way, on YouTube. I'll try to answer them. I'm gonna be checking time to time YouTube and the YouTube chat to see if there are any questions. Actually, in fact, let me open up it as well again, so that it's on the side before we start. Okay. All right, then I guess we can get started. So welcome, good morning again, and welcome everyone to our first lecture. So we're gonna be covering the basics of the PNS Accelerating Genomics course. We're gonna be introducing what this course is going to include and what we're gonna expect from students and so on. So let's get started. So this semester, we're offering two PNS genomics courses. So first one is a PNS Accelerating Genomics course, which is this one. And this course, we're going to be focusing on mainly on co-designing hardware and software to accelerate genome analysis pipeline. And we also have a PNS Mobile Genomics course. And in this course, we mainly focus on designing hardware and or software mainly for constrained computational resources. For example, low power design so that they can work on, for example, mobile devices, right? But in general, both of these courses are project-based courses. And these courses are mainly taken by bachelor's or master's students. We're going to be having weekly lectures on the basics of genomics. And the students are going to be having an hands-on research exploration with the mentors that we're going to be assigning to them. They're going to, by doing so, by following the lectures and the projects, they're going to be improving their research and presentation skills throughout this semester. So this is basically how the rest of the semester looks like in terms of the weekly lectures. So we're going to be offering the weekly lectures on YouTube. And this is a tentative schedule. You can find the schedule on the course website. So I'm going to be coming back to the weekly lectures in the next slides again. So we offered this course, the PNS Accelerating Genomics course last semester as well. If you're curious what we have covered in the previous semester, you can check our course website from last semester. And you can also watch the lectures that we released again last semester, which is fully available on YouTube. And this is the link. This one is the link to the playlist of the lectures that we offered last semester. So you can basically check them and then see what we have already offered last semester. So we have been offering the PNS Accelerating Genomics for some basically quite a while. I guess like since fall 2020, and we really had, I guess like good success in the projects that we've been offering and the progress that the students showed. And in fact, actually, some of the projects, some of the students continued working with us after completing their PNS courses. So this is something that you can always keep in mind if you're happy, let's say, with your project. Or if you're basically, if you're interested in joining us and working with us, you can always contact us during the semester or at the end of the semester, showing your interest. And we're going to be basically happy. We're going to happy to consider you, of course, assigning you other projects or letting you to work on the project that you're going to be working on this semester. So with that said, so let's start covering the role of this course. So what is the role of this course? What we're essentially aiming? So we have mainly two categories or two items that you're going to be seeing throughout this course regularly. First one is we're going to be offering weekly lectures, and these lectures are going to be covering the basics of genome analysis, so that students can understand the basics of computational steps of the entire genome analysis pipeline. But they're going to be learning the problems in the genome analysis and how these problems are solved with, let's say, clever techniques, and how the genome is analyzed by following these lectures. The second part is, which is the most important part, is the students are going to be doing hands-on projects with mentors. So by especially at the beginning of their projects, they're going to be learning about the existing efforts, like prior works in the field. For example, for accelerating one or more of these genome analysis steps, right? So it's really important to learn about the prior work so that you can learn or you can understand what is the remaining problem and why you're working on your existing projects, so that hopefully with the aim to solve the problem that still remains from the prior or existing work, right? So these are the two main items that we have in the project and seminar course. So what are the key objectives that we hope that the students are going to achieve by the end of the semester? So hopefully all the students are going to improve their basic knowledge in genome analysis, which is done in dry lab using computers, not the wet lab part. Because they're going to be following the lectures, they're going to be working on a project. So hopefully eventually they're going to learn more about genome analysis. And since they're going to be working on a project and perhaps like doing some coding, they're going to be also improving their technical skills. And if they are working on a hardware, they're going to be also improving their skills in computer architecture. So students are going to be meeting with their mentors regularly. So by doing so, hopefully they're going to be asking questions and discussing with their mentors. So this means that they're going to improve their critical thinking and the analysis skills by meeting with their mentors. So the projects mostly require getting to know about the prior work. So by doing so, the students are going to get familiar with the key research directions in the field. So what people are working on mostly, what are the important problems in the genome analysis field, and what are the remaining problems, like open problems in the field. And lastly, students are going to be presenting their progress and their project to their colleagues. So by doing so, they're going to be also improving their technical presentation skills by presenting their projects. So with all of these, hopefully by achieving the objectives of the course, we really hope that the students by the end of the semester will learn how to efficiently implement one of the key steps. So they're going to learn the key steps in genome analysis, and then they're going to learn how to efficiently implement one of the key steps in genome analysis on a suitable hardware, right? So the suitable hardware part is also critical. So knowing which hardware is suitable is also another skill, right? So you should really know the requirements of hardware, for example, processing in memory, FPGA, or GPU, so that you can answer the question whether that hardware is suitable or not. So, all right, then let's, so let me first check if there are any questions so far. I guess no. Okay. So what are the prerequisites of this course? What we expect from students during the semester and before they start the semester? So we don't really expect students to have some prior knowledge in genome analysis. If you follow our weekly lectures, it's going to be basically good enough for students to learn about the basics of genome analysis throughout the semester. There's not really a requirement for you to have that knowledge. But what we really require is that you should really have an interest in making things efficient. And you should have an interest in solving problems. So these are really the important parts in the project and semester courses. Because you're going to be working on a project, and we really want you to be interested in the project that you're working on so that you can show some progress, of course. We also expect you to have a basic knowledge in C or C++ programming language. You don't really have to be an expert, but it's really good that if you have a basic knowledge in them. So it's really good also if you have taken the digital design and computer architecture course or somewhat equivalent, so that also you know the basics of computer architecture, let's say. We offer this DDCA course this semester, which is offered by Professor Onur Mutlu, and these are on YouTube, so you can also follow these lectures if you want. So it's really plus if you already have an experience in FPGA or GPU programming, so that we can consider perhaps assigning you to more relevant projects. So these are more or less the prerequisites of the course. So what are the requirements and the expectations so that you can be successful in this course? As we said, students are going to be carrying out a hands-on project. This means that they're going to be building, implementing, coding, and designing, let's say, the solutions that they believe that they can solve the project or that are basically in line with the projects offered by mentors. And you should be doing this with a close engagement with the mentors, with your mentors, essentially. So because like they're going to be assigning you tasks, they're going to be telling you essentially what the next steps are for your project so that you can start working on them. So to do so, and also to show some progress, you should be meeting regularly with your mentor. So this is really important. We also expect you to show some participation. For example, you should really follow the lectures that we're offering weekly so that you can understand the basics of genome analysis. Otherwise, in my opinion, it's really challenging for you to work on a project without really understanding what's going on a high level, right? So it's really important if you can understand why we're doing all these steps or why we're basically pursuing that project. And it's mainly possible. Of course, it's important that you're going to be doing the task that your mentor is assigning you, but it's also important for you to follow the lectures so that you can have a basic understanding of what's going on. You should also, of course, meet your mentors, and you should participate in these meetings, meaning you should basically ask questions or contribute to these meetings with your thoughts and ideas so that you can have some intellectual conversation with your mentors. And your mentors are going to be assigning you some materials such as papers for you to read so that you can learn more about that project and also the prior work. So you should really read those materials that your mentors are going to be assigning you. So at the end of the semester, we're going to expect you to present your project and your progress. If you did a coding project, you're going to be also preparing a GitHub repository where you're going to include all your code and all the other materials in the repository, and then you're going to be delivering that repository to us as well. So if you've really done, if you've really shown a good, let's say, results and good progress, and if you believe that the progress that you showed and the results that you showed are publishable in some venue, we're going to be telling you that. Actually, we're going to be telling that opportunity to you. And if you're interested, we're going to basically help the projects with good progress to get published in good venues, good conference venues, or in good journals. So keep that in mind as well, right? So if you're really interested, you can be publishing the project that you're working on. So then, what are your responsibilities to have basically to show some successful progress in the semester? So as I said, you should be following the lectures, right, regularly. The schedule is on the course website. We already put the entire tentative schedule. It's subject to change, but it more or less shows what we're going to be covering in the semester. So you should be working on your project regularly. On average, perhaps like 6 hours per week. But of course, like we know that this is not always possible to work 6 hours. Like sometimes it's going to be perhaps less than 6 hours, maybe more than 6 hours. But it's important that you work on your project regularly, and so that you can show some regular progress to your mentor, right? You should also be meeting with your mentors regularly. And the last thing is that you should be following, for example, regularly the course website, the Moodle page, Ring Central, and your emails, so that you can see the important announcements and the important messages from your mentors. And you should be responding your mentors timely, so that you can plan the rest of the semester well with your mentor. So we've already have 2 assignments for you, mainly 2 assignments. One, I guess, which is already is something you already know, is we sent you Google Forms and asked basic information about yourself and your skills, so that we can assign the projects to you based on your skills and your interest. So this is really important. So if you haven't done it yet, please do it, so that we can assign the projects to you more efficiently and let's say effectively for both you and also for the mentors. So the second assignment is we've put this paper on the course website. You can, I guess, see it in the required materials on the website. So it's really important for you to read this paper. This is published last year, and this paper really covers almost entire steps in genome analysis, starting from actually library preparation and sequencing from the wet lab down to the variant calling step, which essentially finds, let's say, differences between an individual compared to the entire population, so that, for example, you can pinpoint the mutations and associate them with diseases, let's say. So if you read this paper, hopefully you're gonna have some high-level understanding of more or less the entire genome analysis pipeline, so that you can see the entire picture. And in my opinion, it's important to see the general picture first, so that you can start basically you can start understanding basically what's going on in the coming lectures. So that's why it is a required reading, and we expect you to read it before the next lecture, essentially. So with that said, let's start covering the topics related to project assignments. So as you know, we're going to be meeting today in person, and the mentors are going to be explaining the project that they're offering to you. They're going to be explaining details about the projects, and they're going to be taking your questions about the projects or the course itself. So it's really important for you to attend this meeting, either in person. We've already released where this meeting is going to be in person, or on Zoom, or you already know the Zoom link as well to this meeting, or you can watch the recording of the meeting. So we're going to be recording the meeting, and then we're going to release the recording of that meeting later on. We also expect you to complete the assignments 1 and 2, so that we can make a good project assignments, right? We can assign so that we can assign the projects to you in a good way. And finally, we're going to ask you to select the type 5 projects that you want to work on, given that you already seen the projects, right? Given that the mentors already explained the projects to you. You're going to be able to select the type 5 projects on Moodle. We're going to create a form or poll on the Moodle page, starting from tomorrow. I assume it's still subject to change, and we're going to announce them, but you should expect that the project assignments should start tomorrow. And the deadline to provide your preferences is March 8th. I assume it's Wednesday. So basically, please attend or watch the recording of that meeting we're going to have about the projects, so that you can make an informed decision about the projects that you want to work on, right? And finally, after having your preferences, we're going to match your interests with or basic skills and background with a suitable project, hopefully, and then we're going to discuss it with you. So after assigning the project to you, we're going to put you in touch with a mentor, so that you can start planning the rest of the weeks with your mentor to start working on the project. So then let's cover like who are we, who are the mentors, because they're going to be offering the projects to you. So it's important to get to know them. So let me start with myself. I am Can Fırtına. I'm a PhD student in the Safari research group. And my research interests broadly span bioinformatics, real-time genome analysis, genome editing, and the hardware acceleration of genome analysis. So you can get to know us and our research visiting our website, website of our group, or you can contact me using my Gmail or my ETH email that you already have. Or if you want to learn more about the research that I'm doing, you can visit my academic website. And this is my Twitter account. So we also have several mentors, and whom some of them are basically senior researchers in our group. For example, Mohammed Alser is a senior researcher and lecturer in our group, and who is also is a mentor in this course. We also have Juan, who is also a senior researcher and lecturer. And we also have Hayu, again, who is a senior researcher in our group. So you can see their research interests over here, and you can actually visit our website over there to learn more about them, to learn more about their research interests. We also have some PhD students as your mentors. We already covered me, I guess, so I'm going to skip. And we also have Nika, who is a PhD student in our group. We also have Joel, again, a PhD student in our group as well. We also have some master's students who, again, can be their mentors, can be your mentors. For example, we have Arvid, we have Julian, and we also have Max. So we also have Banu as one of our master students, as one of your potential mentors. And we also have Yunju and Felipe as visiting students. Yunju is also a mobility student in our group. So it's important to get to know them because some of them are going to be your mentors in this semester. So the projects we're offering this semester, in a high level, they all aim to basically achieve intelligent genome analysis. But then the question is like, what is really intelligent genome analysis, right? So each of these projects that we're offering have a certain direction. They are all in a basically cover a wide spectrum of problems, let's say. And each of them basically aims to solve one of these main directions in intelligent genome analysis. And these directions are mainly fivefold. First, to achieve an intelligent genome analysis, we really want to basically perform fast genome analysis, right? Even perhaps like real-time genome analysis, because we want the answers from genome analysis as quick as possible, because they can be life-critical decisions, right? So it's really important to achieve a fast genome analysis. So the second is we really want to utilize the architecture that we have today so that we can perform both in energy efficient and low latency genome analysis using the suitable hardware. For example, like we can use a certain hardware which can reduce the data movement, which then eventually improves the entire performance and the energy efficiency of the genome analysis step. The second is we know that the DNA is a valuable asset, so it's really important to keep in mind that the DNA data is secure and private. So another point is that it's really important to achieve a good scalability in the genome analysis, because we're not always basically doing genome analysis for an individual, but sometimes we're doing a population scale genome analysis where we have, let's say, in our sample where we have basic DNA from many people, let's say, from thousands of people or thousands of organisms. So it's really important to be able to perform a scalable genome analysis, right? If you want to basically, for example, perform genome analysis anywhere at large scale. And another point, which is perhaps like one of the most important points, is that we want to basically avoid erroneous analysis, right? Because we don't really want to, so it's of course important to perform fast genome analysis, but eventually what's important is that your answer to that genome analysis is correct, right? If you're giving an incorrect answer, even though you're doing it really fast, let's say, it's useless. So it's really, really important to keep in mind that the results of the genome analysis are accurate. For example, we don't really want to diagnose someone with an incorrect disease, which could be disastrous for that person, right? So these are basically mainly the directions that we're aiming to achieve so that we can achieve intelligent genome analysis overall. And you're going to see that the projects are mainly about these directions, at least one of these directions or more. So in our next meetings or next week, the lectures, we're going to be covering the entire genome analysis pipeline, starting from, as I said, the sequencing part down to the analysis part, for example, like how we generate the data by preparing the DNA sample and putting the sample into a DNA sequencing machine, which reads your DNA and then generates the output as ATGCs, let's say, the nucleotides of your DNA molecule. And we're going to be basically showing you how you can basically access such data, because they are available in certain databases that can be accessible to the public, for example, NCBI. Or we can perhaps even simulate the data. There are simulators to generate this data. Then basically, there are some steps basically that translates the data that is generated from a sequencing machine to ATGCs, and down the road, like what we do is that we try to construct one's full genome from the data generated from sequencing reads, so that we can do accurate analysis. So we're gonna be covering all these steps in our next weekly lectures. So that's why it's really important for you to follow these lectures. So let me then show you again the tentative schedule that we're going to be covering this semester. So we're going to be essentially starting with the basics of genome analysis. So we're already covering the first one, which is the introduction to this course. It's not really including the basics of genome analysis, but starting from next semester, we're going to be seeing the basics again, genome analysis. And in the next lectures, so we're going to be basically going a little bit specific, and I'm talking about the solutions to address the important problems in one of these steps that we cover in the early weeks, right? So we're going to be basically talking about the papers or the works that are released, published by our group, and perhaps like other groups, so that you can learn what the state of the art is in the field. So if you want to learn more about genome analysis, especially learn more about one of the early steps in genome analysis, I really suggest you to read this paper. So this paper really covers almost everything, let's say, from the early stages of genome analysis, how people achieved or aimed to match the DNA sequencing data generated from the sequencers to either reference genomes, basically a representative of a certain species, or to basically reconstruct one's genome. So basically, you can see all the basic steps that have been taken since, I guess, 1980s up to today. So it's really a comprehensive paper, and you can really see the history of basically how the algorithms in genome analysis evolved over time. So we also have some content that covers how certain suitable hardware can be used to accelerate the genome analysis, right? So if you're interested in learning those, you can read this paper. We also have another paper, again, like covers how basic genome analysis can be done using near memory architectures. We also have Genome, which accelerates the approximate string matching, again, on a near memory device. We also have GenStore, which basically uses SSDs, the flash storage or in storage, basically, so that we don't really move the data from the storage from the disk to memory, because we can do some of the analysis in the storage as well. This means that we reduce data movement significantly, which then eventually reduces the energy usage and improves the performance of the entire genome analysis. We also have this recent paper, which was presented in Micro last year. This paper basically uses an in-memory acceleration to perform genome analysis, let's say, in memory, by coupling two important steps in the genome analysis pipeline. So this is another work that was presented in ISCA Segram. This is essentially the first work that uses graphs to accelerate the genome analysis, so that we can perform genome analysis using graphs, right, using a hardware acceleration. So there are also other works. For example, this work uses, again, in-memory computing to perform fast genomic sequence identification, let's say similarity identification. So there is another work. This basically work uses the real in-memory device to perform sequence alignment, again, one of the steps in genome analysis. We also have the other works that, for example, uses hyperdimensional computing for microbiome profiling. We have Sneaky Snake that can use either CPUs, GPUs, or FPGAs to perform quick filtration of the sequences, DNA sequences that are not really useful, let's say in genome analysis pipeline. Gatekeeper, again, is a similar work to Sneaky Snake, is an earlier version, let's say, of Sneaky Snake, or prior work from Sneaky Snake. We also have like other software-focused works. For example, Blend can find approximate matches with an instant, basically, computational complexity, almost instantly, without really using computationally costly algorithms. We also have Airlift that can map the reads from one reference genome to another one if these reference genomes are close to each other. FastRemap, again, is similar to Airlift, which is a more simplified version of Airlift to basically perform the most important steps in remapping. We also have RowHash, which can quickly analyze the nanopore raw signals as they are being generated in real time while reading the DNA, so that these raw signals can be mapped to a reference genome in real time while the data is being sequenced. So if you want to learn basically more about accelerating genomics, you can watch this lecture. So here's a link. You can also find these slides on the course website. By accessing these slides, you can also access these links. So you can watch this lecture, or you can basically watch this talk, again, if you want to learn more about accelerating genome analysis. We have, again, more talks, such as this one, or again, this one. So these are really important. If you are really curious about this stuff, it's really important to cover these lectures, or for example, on fast genome analysis given by Professor Onur Mutlu. We also have, we've basically given lectures, again, like on genome analysis. You can find all the relevant links and playlists to these lectures here again. So with that, I guess we've covered hopefully almost everything about this course. Hopefully everything is now more clear to you. So let me quickly check if there are any questions on YouTube. If not, we're gonna be, I guess, okay, there are no questions. I assume I'm gonna be waiting for a few seconds, just in case. Okay. I guess there are no questions. So with that, I guess we can stop the live stream and see you in our next meeting or in the next lecture.